a low-power image of a lobule of the pancreas. On the right side you can see a bit of connective tissue septa that would divide this lobule from another lobule. Coming down from the top of the lobule you can see a profile of a large artery and a vein, and almost at the extreme left of the lobule you can see a view of islet cells. We'll be looking at the secretory acinar cells in more detail for this laboratory. On this slightly higher magnification of the pancreas you can see a cluster of islet cells on the left and lots of secretory acini that consists of groups of acinar cells scattered throughout this lobule, and also you can see some profiles of intercalated ducts. We won't concentrate on the ducts for this laboratory as we'll get to that later in the course. On a still higher power view you can see nice clusters of acinar cells with their basal nuclei, and you can get a sense of the zymogen granules, these secretory granules scattered throughout the acinar cells. You can also see a couple of profiles of intercalated ducts, one near the top of the image about at 1 o'clock and a pair of them at the bottom of the image. You can see some very small profiles of capillaries, and you can also see some cells that are called centroacinar cells. There's a pair of centroacinar cells on the left-hand side of the image around 9 o'clock. The centroacinar cells form the smallest divisions of the secretory ducts in the pancreas, and we'll get to these later in the course. Now the zymogen granules are very evident on this preparation, and it's interesting because acinar cells tend to show a apical acidophilia because of the zymogen granules, and more or less a basal basophilia because of the rough ER that is synthesizing the proteins that are in these zymogen granules. Now this is not so evident on this preparation, but it is very evident on this prep, which has been overexposed photographically just to highlight the basal basophilia versus the acidic acidophilia. Now the basophilia, the dark blue staining, is due to the fact that there's a tremendous amount of rough ER in these cells which are actively engaged in protein synthesis. The apical acidophilia is a result of the zymogen granules. These are, of course, the pancreatic digestive enzymes which are packaged into vesicles, and these vesicles are stored until the cell receives a signal for secretion. So the acinar cell is the prototypical example of a protein secretory cell that secretes by regulated secretion. This is an H&E stained image. We'll point out that the dark blue, the basal basophilia, is due to the hematoxylin staining, where the more orange stain is due to the eosin staining components of the dye. The center of the slide is a large profile of an excretory duct from the pancreas surrounded by connective tissue. So really what you're looking at is connective tissue coming right out at you in the plane of the section, with a lobule on the left side again coming down into the plane of section. This is actually a view of a plastic section that's prepared for electron microscopy, but the section is viewed with the light microscope. It highlights nicely some of the basal nuclei in the acinar cells, and you can clearly see the zymogen granules. Up onto the upper right-hand side of this image you can see a profile of a centroacinar cell. And now the term agastroplasm is an old-fashioned term that simply means just rough ER. And on the extreme right, just above the nucleus, you can see a little bit of a clear area that suggests the Golgi apparatus. In this electron micrograph of a portion of an acinar cell, you can clearly see the zymogen granules on the top part of the image and the large amount of rough ER on the bottom part of this image. On this very low power electron micrograph you can see some acinar cells with the basal nuclei. You get a hint of rough ER and then the dark zymogen granules, and you can see the direction of the red arrow. The tip of the red arrow is actually in a canoniculus between acinar cells where the zymogen granules are secreted into. So direction of secretion is in the direction of that red arrow.